Thank you to the organizers for this invitation. So when I was born many years ago, Emil Zucker-Kandel and Linus Pauling published an article entitled Molecules as Documents of Evolutionary History. And that was actually the foundation for molecular phylogeny, and also had a profound impact in many fields of biology, in many areas of biology, the years to come, from molecular systematics to population genetics and comparative genomics. And I would like to talk today, so my talk will be a very general overview about how this molecular information can help to, or has actually helped to uncover, to unveil the real extent of biological diversity, which I will argue is microbial, and how in turn the discovery of this new microbial diversity is helping us to answer questions in ecology and evolution. So the study of microbial diversity has been always linked to the novel technological developments, the first of which was the microscope, which allowed actually to see microbes for the first time at the onset of the 18th century, but then led to the formulation of the cell theory, to the first attempts to reconstruct molecular phylogenies based on morphology and including microbes, and as late as the mid-20th century, to discover that cells are divided in two major structural types, the prokaryotic and the eukaryotic types. By that time, we also had learned that DNA was the storage polymer for genetic information, and also we had known about the DNA structure, and that opened the development of molecular biology in subsequent years that also set the ground for the development of molecular phylogeny. But by that time, the mid-20th century, microbiologists had also realized that morphology has serious limitations and that you cannot distinguish all microbial species based on morphology. There are different variants, but this is very limited information, and you cannot more precisely establish evolutionary relationships between different species. So that as late as in 1963, a famous microbiologist, Roger Staniet, very pessimistically concluded that the ultimate scientific goal of biological classification, that is natural classification, a classification of organisms based on phylogenetic relationships, could not be achieved in the case of bacteria. And actually, microbiologists appear to be condemned to more functional or practical, operational taxonomic schemes where different microbial species were classified according to their phenotypic properties, mostly metabolic properties of microorganisms isolated in clonal pure cultures in the lab. But this was not a natural classification, a classification based on evolutionary relationships among organisms. And of course, things changed radically with the possibility to extract evolutionary information from molecules, because now it would be possible to pick a molecule that is present in all living beings, in all cells, and then by comparing the sequence of that molecule in many different organisms, from bacteria to elephants, you could place them in the same phylogenetic tree. You could compare the same very character. And this is actually what Carl Woese did in the late 1970s by using as a molecular identity marker the ribosomal RNA from the small, the RNA from the small ribosomal subunit, which is, of course, the machine where protein synthesis takes place, and that is universal. And by doing that, actually, Carl Woese was able to do two very important things. One was to actually show that it is possible to reconstruct universal phylogenetic trees by using this type of molecular information, including things like different eukaryotes, animals and plants, but also bacteria. And second thing, and this was an accidental discovery, by doing this type of trees, he realized that there was a third group of organisms that look like bacteria, but that like to thrive in particular extreme environments, in anoxic settings and other weird places, and that detach in that tree, forming an independent group that he called the archaebacteria, and he later reclassified as the third domain of life. So 3 domains of life: bacteria, archaea, and eukarya. Although later we learned that archaebacteria that resemble morphologically bacteria are actually much more alike eukaryotes. So with this type of approach, in addition to make phylogenetic trees, we opened a new possibility that was very, very important to study microbial diversity in the wild. Why? Because microbiologists had known for a very long time that microbial communities or organisms living in natural environments cannot be easily brought into culture in the lab, so that only a tiny fraction of the diversity that is out there can be cultivated in the lab. And so one possibility would be to get these molecular markers directly from these natural communities, sidestepping culture in biases. And this is actually one type of approach that has been extremely successful. And at the beginning, it started like this. You can extract the DNA from natural communities. Your favorite community can be soil, it can be plankton, it can be your gut. And then once you get the DNA, you can amplify this particular marker gene that we use, the 16S ribosomal RNA genes for bacteria and archaea, and you can amplify this using specific primers for bacteria, for archaea. You can clone these amplified fragments and then sequence them by picking individual clones. And once that you sequence them down there, you can compare these sequences with sequences existing in databases, reconstruct phylogenetic trees, including your environmental sequences plus those in the databases, and identify groups of monophyletic groups of sequences that share more than a given percentage of diversity, and define in this way operational taxonomic units that are usually defined at thresholds like 97, 98%. That would be the equivalent of the species level for bacteria and archaea, for instance. So the first applications of this strategy date back to the late 1980s and beginning of the 1990s, and they already give or allow to discover new lineages. Here are 2 examples: the discovery of the Pelagibacter clade, so the SAR11 clade, which is, so Pelagibacter is likely the most abundant genus, the genus that covers most diversity and abundance in oceans. And that is a member of the alpha Proteobacteria, and also the marine archaeal groups 1 and 2. Marine group 1 is now known as the Tomarchaeota, and with years it has turned out to be a key player in the nitrogen cycle in oceans and soils. And with time, with many years, we have accumulated, of course, many more sequences coming from many different environments, and we have even curated devoted databases that curate the sequences and keep only sequences of high quality. This is just one example, Silva, one of these databases that keeps alignments of partial sequences, but also of near full-length genes. We have almost 2 million aligned sequences of very high quality and long sequences, and the discovery rate is amazing. So in 2012, something like 75 of the new entries corresponding to what would be new species, so at this particular threshold, and about 45 of these new entries, these new sequences deposited in the database, would correspond to new genera. And this is without taking into account more modern, let's say, methods like massive amplicon sequencing that are now possible using these high-throughput sequencing techniques. So this is also known as metabarcoding approaches. And here, what you do is just to amplify a smaller fragment from your, I don't know if you see, but anyway, a smaller fragment of your gene marker. This is just because these new sequencing techniques produce shorter reads. and then you can sidestep cloning and directly sequence these amplicons, and in this case produce billions of reads, billions of sequences that of course you cannot analyze by hand now, and you can build up OTUs, operational taxonomic units, by other methods of clustering, not direct phylogenetic analysis. But then once that you have defined these OTUs, you can pick a reference sequence for each OTU, reconstruct phylogenetic trees, and classify those organisms in the environment based on that information. So this is not metagenomics. There is a lot of confusion out there and a lot of misuse of the term metagenomics. This is just amplicon sequencing. There is just one gene, many copies, but just one gene. You can also do metagenomics. And metagenomics consists in the sequencing and analysis of all the genomes, the whole genomes of all the organisms in a given community. So this provides another type of information because you have access to the metabolic potential of natural communities by analyzing protein coding genes. And then you can also analyze metatranscriptomics, so the transcript, the activities, the gene expression of these communities, and even metaproteomics, but that's something much more challenging. So the thing is that most microbial communities out there are really very diverse, and the metagenomes are also very complex. So you will be lucky, so you have access to the potential, metabolic potential of communities, or the functional potential of communities, but it's very difficult to reconstruct, have access to genomes of all the organisms in the community. At most, you can have one genome, or the genome of the dominant organisms, that can be really reconstructed. So there are ways to deconstruct this complexity by, for instance, selecting parts, subsets of the community, or even going to work at a single cell level by, for instance, micromanipulating individual cells, or sorting cells by flow cytometry, fluorescence activated cell sorting, or by micromanipulation. And once that you have these individual cells, then you can perform whole genome or transcriptome amplification and analyze those genomes. So this type of approach has led some people to discover new lineages, and in particular have access to what some people call the microbial dark matter, organisms, so to gain information from organisms that represent new big taxa that are out there and for which we lack culture representatives in the lab. So that these lineages would be equivalent to phyla, so that is what we call candidate divisions, but they are not very abundant in nature, and so they are more difficult to access. So single-cell genomes are still, in general, very partial. They are not completely free of problems, because sometimes you can have contamination, but yet there is an interesting new information coming from these approaches. As I said, you can also, in some cases, reconstruct genomes directly from metagenomes by binning approaches, and this has also led to the discovery of new groups of bacteria in this case. So there are several groups of, actually a whole diversity of clades of new bacteria with unusual biology that had escaped previous scientific detection simply because they have divergent ribosomes. So the markers are very divergent that you wouldn't pick them with universal, so-called universal primers for bacteria. And in this case, these particular groups of bacteria have been detected in the subsurface, so below ground, and many of them correspond to cells that are really very small. So they are at the limit of what an organism must contain to be really autonomous, so really tiny cells. So with all this information, we are getting a very different picture of the tree of life, where most of the clades in bacteria and archaea lack actually culture members in the lab, and we have very little knowledge about their biology. And these are all the clades that have here a red spot, so they really dominate. And even in some cases, there might be, like this candidate phyla radiation, this can be, perhaps there can be problems of phylogenetic reconstruction because these lineages evolve very fast, but their diversity is real. So this is something to take into account. Now, what about eukaryotes? Actually, the use of these molecular surveys to examine the diversity and characterize the diversity of eukaryotes have been or were quite delayed as compared to bacteria and archaea, simply for the reason that many of these microorganisms, microbial lineages, have conspicuous morphologies, and they could be classified by protistologists using morphology and other criteria. However, we had also known for a long time that there were cryptic species, in particular for protists of very small size with relative simple morphologies. This is just one example, Micromonas pusilla, a picoalga, really tiny, 1 micron in diameter, that is actually a complex of cryptic species that appear to be cosmopolitan, widely distributed. So, the first molecular surveys of microbial eukaryotes, protists, took place in the oceans and at the beginning of the 21st century, and already revealed the occurrence of nucleates such as these groups 1 and 2 of marine alveolates related to dinoflagellates here, and these mast groups, several clades of marine stramenopiles groups in oceans. And there have been many more studies, and now we have even much more massive analysis, such as the analysis carried out during the Tara Oceans expedition that have confirmed and extended those initial observations. And among other things, so they confirmed that the diversity is very abundant, very important, but also that there are some groups that are particularly abundant in oceanic plankton, in particular radiolaria, that Ernst Haeckel had already described very well with beautiful drawings. The MALV, the marine alveolates, diatom, many stramenopiles, and also this group of weird organisms, the diplonemids, which are related to kinetoplastids, that were already described by Lara and co-authors some years earlier. So all in all, with all this information, what we have is that the diversity of the eukaryotic tree looks like this, with several big supergroups, and most of the diversity within the eukaryotic tree is actually microbial, and large multicellular organisms with more or less organized tissues like animals, fungi, brown algae, red algae, green algae, and plant occupy actually a limited space within that tree. And another important thing is that multicellularity appears several times independently during eukaryotic evolution. So there are several paths to multicellularity. It's something relatively easy. It's not that complicated. So, and if we add this information to that of prokaryotes, and we have now explored many different kinds of environments for more conventional environments, soils, marine, fresh waters, but also very extreme environments, picture that we have of life on Earth today is that most of this biological diversity is actually microbial, and we have two domains of microbial organisms, the archaea and the bacteria, which are prokaryotic, and they encompass a wide diversity of metabolisms in carbon and energy metabolisms, although their morphology is not very conspicuous, and the eukaryotes, where most of the diversity is again microbial. And this is actually not surprising if you think that for most of Earth's history, so since life appeared like 4 billion years ago, most life has been actually microbial during this long evolutionary time, and that large multicellular organisms such as plants and animals appear relatively late, 500, 700 million years ago only. So there was a huge evolutionary time, and that explains why we have this genetic diversity that is so wide in the case of different microorganisms. So I would like now to give some examples of how the discovery of this new diversity or microbial diversity, the exploration of microbial diversity can contribute to solve some evolutionary questions. And this is particularly true for the case of microevolutionary questions, historical questions, because molecular phylogenists know very well that increasing taxonomic sampling improves very much phylogenetic reconstructions. So the first example that I want to mention is this one. And actually one of the first outcomes of molecular phylogeny in the late 70s, which was the demonstration that eukaryotic organelles, mitochondria, and chloroplasts derive from ancient symbiotic bacteria, alpha proteobacteria in the case of mitochondria, and cyanobacteria in the case of photosynthetic eukaryotes. But what is much less known is the nature, in particular, I'm going to talk about the cyanobacteria, the nature of the ancestral cyanobacterium that gave rise to the chloroplast in primary photosynthetic eukaryotes. And this is an ongoing debate and in the recent literature. So you have some authors that propose that the chloroplast emerged from a single cyanobacterium early within the cyanobacterial tree. And so, the yellow clades correspond to primary photosynthetic eukaryotes, red algae, glaucophyte land plants, and green algae. But in this case, on the left, there was not any particular cyanobacterial relative that appears close to the chloroplast, while other authors propose that the ancestor of chloroplast was a derived cyanobacterium that was nitrogen fixing, likely, and related to a complex filamentous cyanobacteria. So we have had the chance in the lab to sequence the genome of a new cyanobacterium, Gliomargarita lithophora, that we isolated from microbialites coming from a Mexican lake, and that is the only culture representative of a new lineage of cyanobacteria that is widespread in biofilms of freshwater systems and also terrestrial ecosystems. And it turns out that if you make phylogenomic analysis using many plastid markers, but also markers of, but also genes that have been transferred to the nucleus of primary photosynthetic eukaryotes, this Gliomargarita lineage places as the closest cyanobacterial relative of the chloroplast of primary photosynthetic eukaryotes. And now, if you take into account the ecological distribution of this cyanobacterial lineage, which is actually freshwater and terrestrial, and also the ecological distribution of the earliest primary photosynthetic lineages, the earliest branching photosynthetic lineages, it turns out that most likely photosynthesis in eukaryotes emerged in freshwater and terrestrial and/or terrestrial environments, and not in oceans as is currently assumed. Another example that I would like to mention is that of the Lochiarchaeota. This is a group of archaea that was actually that we had known for a long time because we had the sequences. This is a group that is widespread in marine sediments, but also in other settings. But last year, Anja Spank and co-workers succeeded in reconstructing a genome of this Lochiarchaeota from a metagenome of deep sea sediments close to this Loki's castle, where this Lochiarchaeota take the name. And actually, they identify a number of genes that were present in Lochiarchaeota in common with eukaryotes, many more genes than shared with other archaeal groups. And when you use these genes to make phylogenomic analysis, then you realize that eukaryotes are now nested within archaea and have the Lochiarchaeota as their sister groups. So this, of course, has implications for models on the origin of eukaryotes that classically could be defined in two very large groups. Some group of hypotheses that propose that eukaryotes derive from a proto-eukaryotic lineage that was sister to the archaea that at some point acquired the ancestor of mitochondria, and models that propose that eukaryotes derive simply from a symbiosis between members of two primary domains, archaea and bacteria. There was never a third proto-eukaryotic domain. prior to similar or simultaneously to the archaea and bacteria. And actually the discovery of the Lochia archaeota has eliminated this possibility and provides support for a symbiotic origin of eukaryotes, because now the archaeal component of eukaryotes is really well nested within the archaea. And of course, there are variants of this model. I'm not going to enter into the details. And now I would like also to comment on how the possibility to characterize microbial community in natural environments, and in particular thanks to these high-throughput techniques, to these massive sequencing techniques, is now opening the possibility to answer ecological questions. And in a sense, the questions that were before somehow forbidden for microbial ecology. So in a sense, this is contributing to bring microbial ecology to classical ecological theory. So this is an exciting time for merging microbial ecology and classical ecology. So of course, many of these questions relate at the beginning with microbial biogeography, that of course we must assume that they respond to the same universal processes, mutation, migration, extinction, and drift, that we tend to call, in the case of microbial communities, diversification, dispersal, selection, and drift. But perhaps the relative proportion of these different factors is not exactly the same as for animals and plants, and the patterns may be sometimes a bit different. That is the case, for instance, for rank abundance curves, so curves where you distribute your OTUs, your tags, according to the relative abundance, and these type of curves have much longer tails in the case of microorganisms or the microbial communities than in the case of macroorganisms. This very long tail is very often called the rare biosphere. It might be that some part of these sequences here correspond to artifacts and chimeras, but we are learning to get rid of those. But part of this long tail corresponds to low abundance organisms that are active, but many, perhaps most of these very long tail correspond to inactive, dormant organisms that are produced locally or are dispersing or on the way to decay. And actually, dormancy is a key property in the microbial world. So many microbes, or most of them actually, can produce resistance forms like spores or cysts, or simply reduce their metabolic activity to enter this type of dormant state and remain silent or inactive for days, weeks, months, years, and in some cases of natural long-term preservation methods, such as cryopreservation, you freeze the microbes, or lyophilization, desiccation, even you can have preservation for even hundreds or thousands of years. So this is important because on the one hand, this advocates for the integration of temporal scales when analyzing microbial biogeography. And second, this is also important because it implies that microbial communities have a high resilience, so they are highly resilient. And this property can also explain the traditional view or model of microbial biogeography that is based on the famous tenet by the Delft School of Microbiology, this everything is everywhere but the environment select, which postulates that because microbes have large population sizes, but mostly because they don't find barriers to dispersal and they are very resistant, so they can resist for many time, microbial species are cosmopolitan, which translates at the level of microbial communities, at the level of communities, in the fact that environmental selection really drives community composition. So we would be in a situation where the genetic distance at the level of species and community dissimilarity don't change with the geographic distance. So this is the traditional view in microbiology. But there are more and more claims that microbes, or at least some of them, could also behave like more classical plant and animal communities where endemisms exist with allopatric speciation, and geographic distance at the level of communities can really determine community composition. So in this case, we would have the genetic distance, and also community dissimilarity will increase with geographic distance. Of course, there is an open debate, and you could imagine that there would be a situation where this type of situation would be most favored, like in cases where global dispersal is facilitated by current circulations, such as in oceans, and that that other particular case would be more frequent in environments that are more confined, like islands or lakes. So there are many studies ongoing trying to tell apart one or the others and really describe the real situation. We have also contributed to some of them, and so this is a study that we did on 4 different shallow lakes plus a brook, the Rue Sainte-Anne, located close to one another, between 2 and 9 kilometers away, in this Parc Naturel Régional de la Haute-Vallée de Chevreuse, close to Paris. And we studied here protists smaller than 5 microns that should behave like bacteria, like this, everything is everywhere, but the environment selects. And these 5 different ecosystems have very different, very contrasted physicochemical features, as you can see there. So, and actually we carried out molecular surveys over 24 consecutive months using metabarcoding approaches. And what we could see is that actually these 5 systems, despite the fact they are very close in the environment, they are really different. So there are only 50 OTUs that are shared at some point by the 5 different communities from out of around 4,000. So 5 very contrasted communities, suggesting that really environmental conditions are the main drivers of these particular communities. But despite this difference, they all show the same marked seasonality that you can see when you compare pairs of distances between pairs of communities, pairs of samples, separated by 1 month, 2 months, 4 months, etc., up to 12 months. And then you can see that samples separated by 6 months, like summer and winter, are more dissimilar than samples separating by 12 months. There appears to be an exception here, but this is actually a draft event. And this was interesting because this allowed us to observe how microbial communities respond to environmental disturbance. Because so here you have the 24 months and the different the distribution of these different OTUs in big taxonomy groups, and the bars correspond to months where these communities, these ecosystems, these 2 ecosystems simply desiccated. So we analyzed the diversity also in the sediment that was dry after the water evaporated, and of course, we saw that these communities, so the microbial diversity in the dry sediment was very different from that in the water, that's for sure. There was not a single OTU that was shared, so we could not distinguish even dormant organisms with the number of sequences that we had, and these were several thousands of sequences. But when you analyze or compare communities that have developed in plankton once the water has, after water refill, what you see is that even in 1 month, you have communities that look like the water communities prior to the desiccation of the system. So this indicates a very rapid resilience of these ecosystems, even after 5 months of desiccation. And the same was the case with the other system. And finally, taking now into account the complete survey over the 24 months, we could also have a look into the different big groups like these two groups of algae, cryptophytes, chrysophytes, and the impression that you get is that the dynamic of the different taxa is a little bit chaotic, and actually this reflects very well the individual behavior of each OTU, of each species, if you want. So you have OTUs that peak twice in the consecutive years in winters, others that do the same in summer periods. You have OTUs in the middle down that peak once in one ecosystem. You have other OTUs like on the right down, where you have one OTU that peaks in one ecosystem one year, the next year in another one, whereas in a third one, the OTU is present all over the year. So very different behaviors. So this looks like a bit chaotic. But when you distribute your 4,000 OTUs in very big functional categories, very broad, really broad categories like primary producers, free heterotrophs, and parasites, you can observe a remarkable proportion or proportionality between primary producers, free heterotrophs, and parasites. In the four shallow lakes, the situation is a little bit different in the brooks and tan in yellow because the primary producers are lower. And this is actually can be explained by the fact that this is a stream, so these are running waters, and in this particular system, most primary production is benthic. So the idea here is that, that despite all this diversity and dynamics of individual taxa, there is functional redundancy, and there seems to be a particular load for these three functional categories in these ecosystems. And with that, I come to my last slide to just concluding by insisting on the importance that has had to us, biologists in general, the possibility to extract this evolutionary information from molecules, because we have now a tool to explore microbial diversity for real and microbial communities, and to really answer questions about ecology and evolution. And with that, I want to thank people of my team, Aurelian is here around, and collaborators for long-term discussions and fun doing work, and to you for your attention. Thank you very much.